So hello guys, do you hear me? Here we have Christos, Vadym,
Hello.
and Victor on the call.
Hello.
Hello.
Hi.
Can
So,
you hear me okay?
But there are 3 participation too.
Yes, and we have 3 participation in here in Zoom, and we have how much? 31 participant in YouTube. So, ссылку в чат. Participants in YouTube may switch window and go to the link in comments. It will be right now. And so we have 2 short lectures, one from Christos and one from Vadym. And then we will have a discussion of these lectures and very interesting topics, how we can translate seizures from humans to animal models and from animal models to humans. So I think we can start here. And who will be the first speakers?
I'm happy to start the discussion.
Okay, and do you have a presentation?
I do have a couple of slides to discuss. It's gonna be an interactive discussion. So everyone, please feel free to write your questions and chime in with your comments. It's gonna be an open discussion between everyone, and thank you everyone for joining the session today. It's a great pleasure to be in this conference, and congratulations to the team for putting together an excellent program. I'm Christos Lisgaras. I'm a postdoctoral fellow at the New York University and the Nathan Kline Institute, and I'm working on epilepsy research since my undergraduate studies. And right now I'm using animal models, and I'm looking at seizures, and I'm trying to find ways to stop the seizures using optogenetics and other tools. So Vadym, do you want to introduce yourself?
Yes. Hello, everybody. It's very nice conference, very nice talk. Now we should be careful how our presentations looks like after this nice design overview, which fonts and figures we should use. So very nice. I'm Vadym Gnatkovsky, and I think we can we activate our videos as well?
Yeah, you can.
Yeah, it's okay now. Yeah, I see. Okay, super.
Cool.
Now we can see each other. Hi again. And thank you for organizing this event, and thanks for the invitation to do this brainstorming session. We actually have a small experience how to run these sessions before, and it was always good to have kind of vivid discussions. So as I understand, but very small organization question, that it would be possible to have a link to other people from like YouTube followers to get in the Zoom session as we're doing now. Is it correct?
Yes, now we will post the link. I think we need 5 seconds.
Yeah, super. So yeah,
yes, you can start the presentation, and we send a link to this Zoom webinar on YouTube.
To other people.
Yes.
Yeah, because as Christos says, should be really like informal discussion. So not real presentation, more like we can put some highlights of the problem that every one of us can appreciate and discuss, and then probably the best would be exactly question and answers and ideas of others. So I think Christos can start, and yeah, we will see how it's going, how it will go.
Sure. Can you see my screen?
Yes, it's okay.
Yeah.
All right. So today, as Vadym already said, we're going to talk about seizures in humans and in animals. And I think the best example is probably this slide. It's from the Journal of Neurophysiology paper, where you can see that seizures are very similar between humans, animals, and in vitro. And I think that's very interesting, because that tells us that there is some commonalities between what we see in humans recorded with depth electrodes and what we actually see in vivo in rats and in vitro in brain slices. And also, what is very interesting, I can show you some examples from mice where you can also see this kind of patterns. So this is some recordings that I did in the lab, where you can actually see from spontaneously seizing mice. So I make the mice epileptic using kynic acid. I inject it in the hippocampus, and after a couple of weeks, I was able to record different seizure onset patterns, which is very similar to what we saw before in the slide from the review paper. That tells us a very important point, that we can actually reproduce what we see in humans in slices and in animals, which is very important for moving basic science research into a clinical perspective. So that's the whole concept of the session today. We're gonna talk about the commonalities, but also the differences between what we see in humans and what we see in animals. So Vadym, what is your take on the different seizure onset patterns and how well they can actually be reproduced in humans, but also in slices and animal models?
Yes, thank you, Christos. So it's a very good start, and I would like also to share some points, and it will be really in line what you say. So can I also share my screen now, or you need to stop?
I stopped mine, so you will be able to share.
Let's try it. Okay, and yes, just a couple of slides, and we can start also. Yeah. Well, that's actually. Did you see my screen now? Okay, I have just a black screen. Okay. So exactly as Krista said, the idea is just. I would like to say that we're talking about our topic is electrophysiology at the moment, but of course during discussions, please keep in mind that any questions about how we can translate research from animal models or animal research to the human, to the clinic, and vice versa. So they probably appreciate it, as probably we have another experts in the field that can also contribute. So from genetic, anything else that could be in topic. But let's go with the neurophysiology at the moment. So as Christos said, the point is why we're actually talking about translation from animal models to the human and back. It's because probably we can combine, and it would give us a possibility to combine different techniques that we cannot use in human at the moment. And what I mean by that is if we look at the technique that we use in the patients now, like MRT, scalpel, MEG, microelectrodes, microelectrodes, and also intracranial electrodes. And of course, there is overlap with the technique that we use in animals, like intracranial electrodes in animals, we put in the same way. So we drill holes, we put electrodes inside the brain, and it's completely the same approach that we use now in the intracranial invasive recording in the human, in the patients for candidates for epilepsy surgery. But the beauty of the research that now in animal, we also can do a patch clamp, where we use intracellular recording, we can clamp a single cell, we can do ions recording, different imaging technique, optogenetics. So all these techniques that's available in animals and can be used in the perfect and very good experimental condition, we can actually use these results to translate it to learn some mechanisms of the ectogenesis in the human. But as Christos said, for this we need kind of bridge. So for us as electrophysiology, this bridge is exactly the pattern of the seizures. So what I mean is that we try to discover. So this electrographic trace is intracranial recording from the human brain, and we try to discover different patterns and reply or mimic or model this pattern in the animals with different models, with drug injection or electrical stimulation. So all different models that we have for epilepsy. And we try to reproduce this pattern in the animals. So, and the topic that how we try to translate is kind of, we try to see and compare which pattern representing human and animals, and they discover what cellular mechanisms involved. And in these examples, like, it's important, I think, to just first to speak the same language, right? So in human EEG, we usually talk about low voltage, fast onset, diffuse activity, and so all these patterns can be translated actually in animals as well. So we can set this increase of fast activity at the seizure onset, then it's synchronous activity, bursting activity, and this post-ictal depression, let's say, right? So here I just put an example from different papers. So on top you see the human EEG from intracranial recording from temporal lobe epilepsy, focal epilepsy. And here is 9 examples of all different models. So there's genetic models and some induced epileptic seizures I don't even know, but why I put in it, it's just by the eye, we can kind of appreciate the similarity. And of course, all these seizures looks different, let's say. But if we start to talk about which pattern involved at the different stage of the seizures, we actually can try to find out some similarities. Like some seizures of this recording, like here, for example, there is some fast activity onset. Almost all of them, if you agree, have kind of high amplitude bursting synchronized activity in there.
Vadym, sorry to interrupt. I think you didn't change the slide. I just see the first one. I don't know if it's just
mine. Do you still see it?
I just see the first one.
Oh.
That's the same for others, or just my computer?
Is this the same slide still?
Yes, yes.
Okay, maybe I'll just stop this way and probably, let's see.
Maybe if you share it again. Yeah.
Okay, so is it changed now?
No.
Nope.
No, I think I need to start sharing again. Probably. Okay, I'm stop share. I will try to share it again. Probably, if it will work. So.
Yeah, I can see it now.
So is it human brain and the mouse at the moment, right?
Yes.
Yeah.
Yes.
And you've seen this slide already?
Sure. Yeah.
Okay. So now we move to this slide when, as I say, I just pick up 9 different modeled seizures, and to appreciate how and what we can like have a similarity from the human trace and the animal model, and in these 9 different models recorded from the animals. So as I say, you would agree that all seizures looks different, but some patterns, they can be recognized that help us to actually identify they are the seizures. So let's say all of them, if you agree, have this synchronized activity. What is interesting that all of them actually demonstrate this nice post-ictal depression, which also an interesting topic itself. But again, these biomarkers of the ictal event of the focal seizures, they are representing human, and this is the same what we try to figure out in animals. Now, as I say, what is good about have this kind of reproducibility of the pattern of the ictal event in the animals, that we can use different technique now. And this is topic of my next talk. I think it will be on Sunday, but I just show small examples of what can be done. So let's say this is how we approach it in the research. So here on top, we have 2 different seizure patterns from human. Then we have tools to reproduce the similar pattern, let's say, in animals. But now, if we do intracellular recording in animals, we actually can find out which individual cell doing during the seizures. So like in this case, it's increase on interneurons spiking. There is different superficial and deep layer cell participate differently in the cell. And then we can kind of translate this finding back to the human pattern here and discover that probably this low voltage fast onset at the beginning, interneurons is important role on that. Then we can also record that like ions, potassium increase, and all these different techniques can help us to discover what's going on inside the human brain during ictal events. And it's just an example how we can use this kind of workflow from animal to human and back to have an ideas. And I think there's other research about epilepsy do pretty the same thing. So I just would like to invite anybody who would share their experience and their feeling about it. And what the problem we have, right? So from electrophysiological point of view, these kind of things. So we are continuously working to mimic some events and then try to discover what mechanisms producing these events and then to translate and have an idea, have a clue with these events the same in the epileptic patients. And it somehow works up to now for many years. But again, it depends which electrodes we are using, what we're recording. It's important to keep in mind. It's quite difficult to combine different domain of recording. So if you want to do high frequency oscillation, you need to have appropriate electrodes. But this is true for human, but also for animals. So if you use very big electrodes inside of the animal brain, don't expect you will have very nice focal isolated activity. So you need probably high impedance electrodes. And for the units, you need tetrodes or even glass pipettes, but again, depends of your experimental condition. And if you want to analyze, of course, all the network of the brain, like complex epileptogenic network, don't expect you can get it from hippocampal slices, let's say. So all these kind of difference of spatial and temporal domains is important to keep in mind. So I think it was my small point to put on the discussion, Christos. And
yeah, that's great points. And I also think that, you know, depending on what people would like to investigate, they might need to take into consideration which electrodes they want to use, which model they want to use, and also which filters, which sampling rate, which is very important for, you know, uncovering other epileptiform activities like high frequency oscillations that are very important. And yeah, it depends on the study design and what is the question. For the study to choose a model and what you would like actually to investigate. I think the session is open for discussion, so we would be happy to have questions and some points for the discussion.
Yes, now we have a link from Zoom, from YouTube to the Zoom webinar. And then if you in Zoom webinar, you can raise your hands if you participant and you can't turn on the audio and video. So you can raise up the hand and we give you time to speak. And we have several people on call, Victor from our laboratory and Anton from our friendship laboratory, and maybe they have something to say.
I think I have a discussion point about the seizures that we see after brain trauma. Probably Ilya can tell us more about what are the patterns, or if you see something similar from what we showed before in our slides, or anyone with experience with that model.
Yeah. So sorry, can you answer the question again?
Yes, sure. So I was saying about seizures after TBI.
Mm-hmm.
And whether they look like the ones, let me share my screen to show. So they look like the ones that we showed here, or you see something different in the animals?
You know that we have a different seizures in animal models after TBI. We have an experience that animals have an something like absence seizures. So with a long immobility state without any behavioral other abnormalities, without convulsion and so on. And we saw, for example, myoclonic jerks in rats. It's look like a very short, rapid movements of head and maybe upper limbs without a loss of tone. So, you know, the big question that people with post-traumatic epilepsy have focal seizures. But what we see in animals from clinical side, I can't, in some situations, I can't say that it is a focal seizures because the pattern may be in both hemispheres. The clinic may be like absence or like myoclonic. So this is a question, how can we translate seizures, these seizures after focal trauma to humans, because humans don't have any seizures like with generalized onset after trauma, for example. This is a very interesting question for me.
Yeah, that's
regardless trauma and
yeah. And
so,
also, I think what is also interesting is that probably the different seizure onset patterns might respond differently to anti-epileptic drugs. I'm not sure if Vadym, there is a study about that, looking at how anti-epileptic drugs affect different seizure onset patterns. Are you aware of any studies?
I'm not sure. As for my memory, I cannot recall that they really modified the patterns. Usually, of course, people with the research looking for the big things. So they count seizures, and the best thing they can show that when the seizure stops.
Yeah.
And if they not stop, they start to demonstrate whether it decreased the amount of seizures. And they also like to, as I know, count spikes during recording. So if there is, you know, the problem of animal models, of course, is spontaneous seizures we're talking. So
yeah,
it's also already a problem in the human, right? If we want to catch up the seizures, so we record patients for 10 days and we kind of pray for spontaneous seizures in the video monitoring or even in the cranial. With animals, you have the same feeling. So you plant it, you do some model, then you wait, and then you have luck to waiting for spontaneous seizures. And it's not always that you connect many of them. So this is one of the issues, exactly. Therefore, now, of course, with this like improved technique for continuous monitoring, we're happy to have 4 months of EEG recording of the animals. But still not many labs paying attention to this. You know, they kind of, some of them still following this paradigm. So I do some hit, then we wait for 3 weeks, and then we maybe record a couple of hours a day when student or PhD come to the lab, record and go home. So in this 3 and 2 hours, it's very rare that you will be lucky to get spontaneous seizures. And I think this is the way, at least how we're approaching it. We just get this sensor or trying to do as much as long to continuous EEG monitoring to then discover the dynamics, right? Because this at least some what can be used to collect more ECTO events and then measure your pharmaceutical approach or other therapeutic techniques for that. Yeah, I don't know how you experience on.
Yeah, I think that's a great point, because I think the gold standard for defining whether an animal is epileptic and has seizures, I think it's at least 2 weeks of continuous EEG. This is what most laboratories are doing, and so I think it's very important to raise this point, because we cannot be sure if the animal has spontaneous seizures based on scrambled recordings. I think continuous EEG is very important, because seizures might occur during different light phases, during different times of the day, and in females, for instance, might depend on the cycle. And there is also reports about clustering, which is very important. So you might have an animal that doesn't have any seizures for the first week, and then you have a day that has 20 seizures. So this is also happening, and it's very important to be considered when you are looking at the drug effects when you do drug screening. And also animal models have been used in the acute setting. So to trigger epilepsy in the animals, you usually inject a chemoconvulsant, either peripherally or into the brain. So some people are evaluating whether there is an effect of anti-epileptic drugs in this setting, but this might be different from what you get when you have an animal that has spontaneous seizures. So there are different things that you can, extrapolate from drug screening at the acute setting versus when the animal is epileptic and have spontaneous seizures.
Mm-hmm.
Yes, I agree about something like long-term monitoring in animals. So it is very important because, yes, we have to be sure that this animal is epileptic, and to study, for example, epileptogenesis or anti-epileptic drugs, because this is only one way. But it is too hard because animal can't talk to you.
Yeah.
The animal can't say that yesterday I have a seizure, so they can't have a diary of seizures. So it's very hard in animal to conclude that this is an epileptic animal. So I want to ask another question. And what do you think about not convulsive seizures? Because in animals, and a seizure that not accompanied by something like from any motor events. I said about cognitive seizures in humans, about, for example, déjà vu that we have in clinical cases with temporal epilepsy, for example. And people can told you that they have very frequent déjà vu, something like cognitive disturbances and so on, but animal can't. So maybe, and electrophysiological studies may not see these seizures. We can't be sure that we see every seizures on with the, for example, NAG and so on. What do you think about this part of seizures in animals that we can see in humans?
Yeah, I mean, we do see them in the animals. Of course, you don't see any changes in the behavior. So if you don't have the animal with electrodes, you might not be sure that the animal is having something abnormal. But in the EEG, I usually see some changes, and I wouldn't say that the patterns are similar to the ones that I showed before, at least in the intrahippocampal kynic acid model. But for sure, you can see a seizure pattern, a nictal pattern in the EEG, but you might not see anything in the behavior. So yeah, it's a great point, and we don't know whether the networks that support the non-convulsive seizures are the same or different from the ones that are producing the convulsive seizures. But also I think there is some studies saying that non-convulsive seizures can also cause neuronal damage, and that's very important. So yeah, we can get many insights from when we record the animal with electrodes versus just have the animal with video and monitor the behavior.
So yes, but you know, the very big difference between electrographic seizures and patterns and seizures with clinical with any clinical correlate that we can miss in animals. So it's for me it's a very interesting point. Thank you. And we have a lot of new people on the call, so maybe someone want to speak. You can switch on your microphones and feel free to talk something.
Hi. Hello, everyone.
Yeah, hi.
Yeah, you hear me. So thank you for this discussion. I just want to figure out one point. Do we really need to translate to find the same seizure patterns and some EEG signs in animals to make some translational study for humans in future? I mean, the animal brain is on the level of structure, on the conformational level, is far more different from human than if we compare with the level of receptors, genetics, or metabolism. And from my point of view, we need to find some ways to consider the possibility to translate the lower levels like receptors again, genetics or neurotransmission, then trying to reproduce. I mean, reproducing the pattern of seizures in animals, it's really interesting from the fundamental point of view. But does it really have some practical effect?
Yeah, that's a great point, because it's not only that we can see a similar pattern, there might be other mechanisms like genetics or inflammation or other pathways that are affected at the same time in humans. So there is a combination of, let's say, mechanisms that are involved. It's not just that you see a similar pattern, you are gonna say, okay, that's very similar to humans. There is probably other things going on, and it's human is an individual, so there is variability. But the fact that we see similar patterns, it's very encouraging in simulating something that can be helpful. Of course, you cannot simulate everything that is happening in a human epileptic brain versus an animal brain, but it can give you some insight. So if you, for instance, you have some hypothesis that a receptor is important in some kind of seizures, you might want to have an animal that have these seizures, and maybe you have deleted this receptor to see whether this is helpful for the human. So there is some limitations on what you can say, but the fact that we see something similar, it's very important in continuing translational research.
Okay.
I understand very well, Anton, what you're concerned about, this level of complexity of the different complexity from human and animals. But I would say there is still a lot of practical things why we do models, and so because it's all about how we should ask the correct questions, right? So if we want to understand how neurons fire, action potentials like ion concentrations, right? So neurons in animals and neurons in animal brain and neuron in human brain will do pretty the same things, right? From this level, right? Now, if we would like to say seizure generation mechanism, again, if you see that the pattern is the same, whatever you use to induce this pattern. So again, we can learn and study which extracellular ions concentration doing during the seizures, how interneuron, how pyramidal cells firing. So at this complexity level, again, kind of can benefit from each model. Now, if you to know, yes, how the frontal lobe seizures propagate to the temporal lobe seizures and what pathways is doing. See, here probably we are not any longer on the same language with comparing from animal to human. It's probably then computational models, MRI data can be used in something other domains. But this is exactly the topic. So you need to, let's say, researcher, before he do his project, he needs to understand exactly at which level he really can estimate this kind of possibility to translate his data, and where probably he will miss all what you say, other complexity from the entire brain. And what you can do in this hippocampal slices, as I say, is already difficult to translate it to the in vivo animal recordings. So I think it's still a lot of benefits from this, because in human you cannot record potassium or extracellular changes of ions, or do like some microscopy stuff, or like optogenetics research. With animals you can do it easily, and this very basic mechanism of neural activity you actually can translate. So
yeah, but still rodents are more feasible for these basic mechanisms,
right.
But for higher function you need some higher animal primates.
Right. Or models, right? Yeah, or you do or non-invasive fMRI studies, or anything to combine exactly to get data for your hypothesis. That's true.
Yeah. And maybe it was very interesting about propagation of seizure that you told. For example, from frontal lobe to temporal lobe and so on. I think that we can't study it in, for example, rats, because rats have very small brain, and if you saw on the rat brain, it's maybe 1 or 2 centimeters. So to propagate from frontal lobe to temporal lobe in rats, it is take few seconds, but propagation in humans much more longer.
This is very, very correct. And actually, it's actually also very relevant to what's going on in research now. So as you know, the discovery is mTOR pathway. There are so many models now to try to mimic focal cortical malformations.
Yes.
And the people kind of upset because they do this, they see the lesion in the animal's brain, actually a huge one. They didn't do histopathology, and they actually see kind of balloon, non-balloon, dysmorphic neurons, but the animals, difficulty they sees. So here we go. So we have lesion somewhere, and it has all property of the lesion. So we really hit the pathway genetically that's involved in the malformation, and we produce malformation, but we don't see normal seizures as we expect from the human. And this is exactly the point, because so actually I would not be upset by the fact, because these things you cannot easily compare. And if you're talking about seizures now, let's say focal cortical dysplasia in frontal lobe, but it's actually, before it's reached temporal or frontal part, on the semiology point of view, how many times it's like silence? You don't know, you miss an onset from the dysplasia. Then it's spread, then you see semiology, then you discover, then you do surgery. Now, what actually we have, like from seizure pattern from the dysplasia, very few things, even in the human, and translated now from animals, I'm actually happy that they don't see it, because probably they use the same mechanism. But again, we need to keep it in mind. So that's just for the discussion, but it's very important. Exactly, it's not easily to, like, expectation not easily the same as you would hope to get it in the models, and vice versa. Sometimes you have some good results in the models, but then it doesn't work when you try to make your interpretation. I don't know what others think of this.
Sorry. I see next issues in your speech. Epilepsy is a set of different mechanisms, and it is only top of iceberg, and we can translate at all activity, brain activity from mouse or other animals to human, because epileptogenesis is probably same, but the large severe monogenic epilepsy, where only 1 or 2 points are main causes to epilepsy, we can we can retranslate in all for all genes. Don't understand.
Yeah.
Question, please.
Right.
Yeah. I mean, the current models of epilepsy in animals showed only large mechanisms, pro and contra epileptogenesis. Yeah, but in monogenic epilepsies, we can see the main part of epilepsy, like as cause. I mean, S6BP1, KCNT1 gene. If these genes not work as well, yeah, we can see epilepsy, and we can treat target therapy for them. But the more severe epilepsy model, the more earlier onset in this model, the harder create animal models, because mouse died and didn't reproduce such gene models. I mean, that's all.
Yes, it represents a whole complexity of the modeling, especially when you use genetics as well. It's clear, and it's a big topic and concern in research at all, how you model. Sometimes it's easy just to reproduce the pattern, but then how you reach there, you cannot even explain why you have this pattern, right? Like pilocarpine model, there's also many hypotheses why then it's so nice, but it doesn't fit for, let's say, at least some clear pathway connected to epileptogenesis in the human, but it perfectly works. So we use it for many years, and actually for the basic research, it would work. I think way back, it also works in this way, as you say. You do some genetic approach, but animals don't even survive. While in human, everything kind of works. Yeah, but it's a big debate in the research about this. Yeah.
You know, we had a very interesting debate on what is, from 2 points of view, from modeling the seizures and modeling epilepsy in normal animals, and from one side, and from the other side, the modeling of genetic abnormalities, in, for example, in mice, and seizures and this epilepsy with known gene and known mutation, for example. And it was very interesting discussion in our previous seminar. So what do you think about these two ways to model epilepsy in perspectives and more maybe in yes, what do you think from these two way is more perspective to find what is an epilepsy and how to treat it?
I think every model has its limitations. For instance, the examples that I showed you before that were done in the slices, people were using a convalescent to induce the seizures. So they were using 4-AP or low magnesium to reproduce these patterns. Of course, they were using slices from normal rats, but in epilepsy, you might have other structural changes, so you don't really know whether what you see as a mechanism in the slice where you have a normal tissue, it's the same as in the epileptic brain. So of course there is limitations, and we tend to simplify, our model so that we get an answer. But sometimes this might, hinder some studies that have to do with targeting a pathway or a combination of pathways. Of course, I think it's very hard sometimes to get an answer when you add many factors that can change, your outcome. But I think it depends always on the question and what you are trying to study. And then you can choose your, the model or the design of your study based on what you would like to investigate.
Yes, I agree that the aim of study is a main in choosing what model you use.
And you mentioned very also important topic, exactly what we're applying with, actually we're doing it on a healthy animal brain, that if we don't screw up. And I have one nice example. It's also good to discuss, just pointed in discussion from my experience. So I was involved in collaboration to writing a project, a paper. It was about sodium channel mutation in Dravet syndrome. And the model is kind of fantastic, I would say. So in animals, in mouse, the model is if you, so the mutated animals, they burn it nice, they develop, and actually they live pretty well. If you make just one hit with the fan, so increase the temperature when they are very small pups, the animals will cease, have kind of status, yeah? And after this animal will cease once in 24 hours during this transition to the sleep to awakeness. And it will happens every day. So they have this spontaneous seizure, the same as like febrile seizures and Dravet syndrome here. And when we discussed it, it was kind of interesting, because now what happens? So we have this mutation, very precise mutation in this channel in the animals. And they do cease by the fact that there was a hit event and they do it. But this is exactly the point. Now, as you see, they cease exactly more or less in the same time, once a day. So now the brain, health brain, have this problem with sodium channels. But apart of it, other genetic or other problems the brain doesn't have, which could have in real life the patient. And by the nature, the cell or the network was organized by itself just for the circadian rhythms to make just one cease a day. It's like a trigger for the animal model. But this is very important topic, and we always need to keep in mind. So if you really try to touch some mechanisms, keep in mind that all other compensation mechanisms or other probability that genetic or even as electrophysiological mechanisms, they are not in the pathologic brain, they're in the healthy brain, and then your interpretation could be very difficult. That's true, and it's very important point also to keep in mind. Absolutely. But this is all we have for the model, so we're happy at least that we do this research. Yeah.
That's true. So we have several new participants. Maybe somebody want to talk? Please feel free to switch on your microphones or camera.
Yeah.
Yes, for the discussion. We are waiting for you.
Sorry, just shortly, me again. There is a question in YouTube.
Okay.
Are there, from Dmitry Zhuravlev, are there any SUDEP animal models, sudden unexpected death? I don't really know any such a model. I mean, by joke, but at least intentional, I don't know any intentional model. But non-intentional models we have, it's a part of work in animals. Yeah,
I think a mouse model of Dravet has been proposed recently. I think there was a paper in Science Translational Medicine showing that these mice have SUDEP.
You mean SUDEP?
Yes,
SUDEP.
yes, SUDEP.
Okay.
I think the mouse model of Dravet has been proposed, but I'm not sure whether there is a pure, let's say, model that is being used. we know a lot about the mechanism that leads to SUDEP with brainstem seizures and spreading depression. But I'm not quite sure whether there is a specific model that is being used. But I can take a look though.
Okay, and you know, we have another question in our last talk about translation from humans and animal models. What do you think? And maybe it's a topic to discuss. What can we do to unite clinical experience and laboratory experience? So I think that it is a very good, it's a very big problem whole the world, that clinicians is in hospitals and researchers, yes, live in laboratory, and they it's a very few points of
communication between them sometimes.
Yes, yes. So maybe we have an idea how we can unite these two parts of epilepsy world.
I think sessions like this, where we discuss limitations and everything that we are trying to do, would be helpful. But also there are some other summer courses like the San Servolo course, where they are trying to bridge basic and clinical epileptology. So there's a couple of actions and activities that are taking place during conferences and summer courses that can help us to communicate better and try to collaborate more.
There is another problem I saw in sometimes, that clinicians that don't work in laboratory and with animals, they maybe don't want to study these mechanisms, and then can see why they have to study, for example, mechanism and so on. And what do you think we can do? How we can explain to clinicians that studying and learning about animal models, about mechanisms, and so on, is helpful for them?
I feel that the only way to do drug studies before a clinical trial is using animal models. So there is other way. So if you would like to target a mechanism, you cannot do it in humans.
Yes.
So you need an animal model to target and a specific pathway, or brain slices, or other models that are being in use. So I think if you would like to look at the mechanism, there's only one way to go.
I would maybe give an advice in this case. Maybe it might work if you do it by your example. So I would really just build up yourself like a young clinician who involved, who like to be involved in some research projects, and probably publish and do conference presentations. And in this case, just your colleague would look at your career and would maybe appreciate that, clinician who do also interested in basic research or other type of research is not that bad. So like, do good example to your friends, to your colleagues, and it also works well. That's another way how it's hard to convince people, but if you show them that it's actually useful, it helps you to open your mind to be, successful in this case. That could work. Yeah. So
yeah, thank you.
Okay, so it's great advice. And I have other question. I am a specialist in autoimmune epilepsy. So for me, it's very interesting point. For autoimmune epilepsy, we have, for example, NMDA encephalitis with epileptic seizures sometimes, and we have green1 mutation with NMDA receptor, and these children have epilepsy. So we have some axes with mutation in gene, and these are people have epilepsy. And another point, we have antibodies to NMDA receptor, and this patient can have epileptic seizures too. So we can see different problems at different levels in humans. So how we can use it and how we can think about it when we try to perform some translational research. There is some article in Nature about genetic twins, that NMDA encephalitis may be genetic twins of green-1 encephalopathy sometimes. And it's a very interesting point of view for me. So what do you think about it? Can we use this idea for some translational research or something like that?
Yeah, if you would like, for instance, to develop, new therapies, and what is the genetic background that you see in the patients, then you can, generate an animal model and use, some potential drugs that you think might be helpful, and you can see whether this affects, what you see in the EEG or in the tissue. So depending on the question that you have, you can, build the study design that works best for what you would like to do.
Mm-hmm.
If I understood correctly your point.
In general.
Now I think in about some experiment, then you can take on, for example, living slices from rat brains with hippocampal slices and add anti-NMDA antibody to this slice. And no? for example. And do you know we will see any seizures or on the slice electrophysiologically?
Yes, there are a lot of work which discuss this point. There is a lot of experimental model with NMDA antibodies at cell lines or something like that. So for me, it's a really nice idea that different levels of disruption we can see in humans. So we can see genetic anomalies and people with this disease, and we can see some problems at antibodies levels, and we can see this situation in humans. So it's some human, I don't know how to say it. Some models in humans sometimes. Okay, so
yeah,
Christos already told me that we can plan our study and then try to use some ideas, so it's okay. Okay, so maybe we can.
Yeah, there are many different models that people are using. So for instance, people are using the zebrafish model for genetic epilepsies. It's a very high throughput, and other people are using mouse models. So there's a whole bunch of different models that people can consider depending on what the lab and the institute can support. So there are choices out there.
Yeah, okay. Maybe there are other questions. There are no questions on YouTube, and maybe we have some questions in our Zoom webinar.
No, there are one paper from Katya Kobov about SUDEP model, and I think we can send it to Dmitry later.
Yeah.
Yeah, and so we have a lot of people here in Zoom. So maybe somebody want to talk. Yeah, and maybe last question. What do you think we can use in epilepsy models to know something about brain functioning? because if we study epilepsy, it is one way to prepare in the study, but maybe we want to study normal brain, and we can use epilepsy and epilepsy model as instrument to study normal brain and normal functioning, because there are a lot of examples in history and in science that pathology can tell a lot about normal brain. So what do you think about this topic?
I think
we can discuss it. Yes.
Yeah, I think there's a lot of studies out there looking at brain function, perception, and other functions in humans. But these humans have been implanted with electrodes so that you can monitor their seizures. So these are epileptic patients where the data were were collected so that you can uncover a basic function of the brain. So we own a lot to patients with epilepsy, and they are used in different studies to uncover normal brain function. So I think there's many questions about memory, perception, and other functions of the brain that we can answer it through recordings from epileptic patients. And we don't really know whether this can be the same in a healthy brain. Of course, they used recordings from sites that are outside the seizure onset zone. But of course, there is changes in connectivity between different areas of the brain. So you don't really know whether what you see is kind of distorted or not. So yeah, there is some limitations, but we can get a lot of out of the recordings that we do in humans, but also in animals during behavioral tasks, for instance.
Yeah, because the epileptogenesis and epilepsy involved a lot of mechanisms that, for example, neuroinflammation, neuroplasticity, and so on. So we can use this model to study, for example, neuroplasticity, not epilepsy. Yeah. And this is a different aim of study, and so. For me, it's very interesting to work in a lab and to prepare this research, because you can study epilepsy as a disease, and you can study mechanisms, and this is different.
Yeah.
Okay.
Possibly we can discuss another point if we have enough time for this. I mean the models of drug resistance epilepsy, because I see clearly a contradictory point in this, because all models we use are usually drug respondent models, and it is just because we use them as models of epilepsy because they respond to anti-epileptic drugs. And when we try to make drug resistance models from these models, which usually respond well to anti-epileptic drugs, we see that only small % of animals are drug resistant, and I think that it is just a big problem for studying epilepsy, because well, we can treat epilepsy with some drugs we have, but the problem is that we can't treat epilepsy which is resistant to current drugs, and so I think we could introduce some new models which will be focused on drug resistance. And what do you think, which approaches are which approaches could be used to reach this this aim, this goal.
I think it's a tricky point to have, you know, a model that is, you know, purely drug resistant. Of course, you are going to have variability. Some of the animals can be drug resistant, some others might not. And that's an interesting question, why some animals, they are super resistant and other are not. So this is also seen in the human population. Not all the patients are drug resistant. There are some that are responsive. So I'm not sure whether there is a model that is 100% drug resistant in all the animals, but it's an interesting point to, you know, investigate what makes an animal drug resistant, what are, you know, essential so that you have resistance in all the animals that you generate.
Yes, and also this, I think, Christos, I agree with you, but also there is this new model, as I mentioned before, the genetic model for different cortical malformations. And the patient with malformation, they are usually quite drug resistant group, so they are surgery candidates. So it's faulty cortical dysplasia models with different mTOR pathway, genetic mutations, and with electrophoresis. So this model now exists. They are, I think, still in study, but there's already publication that point very well and with good results how to reproduce the pattern, at least in the tissue. And in case of the seizures and the similarity, then it could be also a good candidate for the drug-resistant model to maybe to test some other pharmacological, find out pharmacological pathway to treat these animals, because at the moment this group is the surgery candidates. And I think other models could be developed as well, yeah.
So you see the perspective in introducing new genetic models with known mutations, which lead to known pathology in the brain.
They're very promising, at least, and we know that this group of patients, they're kind of high drug resistance group, no?
Okay, I see, but I think that sometimes in human, the cause of drug resistance is not known. We can't say that it is just because of this malformation. And what do you think about perspectives of studying drug resistance in animals, in model, some kind of animal model, in which the cause of drug resistance is also not known, like in, amygdala kindling, when some percent of animals are resistant to different anti-epileptic drugs. I mean, 30 or 20%, if I remember. So, what do you think about perspectives of animal studies of drug resistance when the cause, the origin of this resistance is still not known? It is not artificially constructed by genetic manipulations, and it is presented just as a phenomenon which origin we do not know, and we can investigate it in animals. But what do you think is will be there a correlation between this resistance in animals and the resistance which is seen in epileptic patients?
It's hard questions. I mean, will be there. The correlation is hard to say, but the fact, I think, that in animal models, you can develop the project that will help you to measure, I don't know, the level of the drugs and see also profile and make a groups which are responder, not responder. I would say it's possible to do it in animal models research. So the direct correlation then with human is a hard story, but if you lower a bit to the basic mechanism of the drug resistance, whether there's ferment problem or some cellular transports problem. So these kind of lower basic biological mechanisms, probably they can be, and I think they are studied in the pharmacokinetics or pharmacology. I don't think that animal models is useless here.
Okay, thank you.
Yeah, I agree with that. I think it's this, the drug resist, the mechanism that leads to drug resistance are very complex. So one way is to, try to find out what are the contribution of specific pathways to drug resistance, but it's not probably be just one pathway. So I think it's a hard project to find out what is going on, because I don't think it's just one pathway that contributes to resistance.
Just maybe we don't have a right drug. I mean, the reason for drug resistance is the same as the reasons for the for epilepsy in partial case. So, and if some epilepsies, some are responsive for modulation of neurotransmission, it doesn't mean that epilepsies originated from other reasons, more complex, should respond to the modulation of neurotransmission, because all the drugs are of the same type, I would say, and we still don't know what is the epilepsy and what exactly should we treat except of neurotransmission.
Yeah, so I agree with this last speak up, yes, and I think we have 2 minutes, and maybe somebody want to join the discussion in this last minutes. If they note, so I think we can go to the end of this session. And so thank you very much for Vadym and Christos to join us in our school, and for the presentation and for this very interesting discussion. And we will waiting for you tomorrow
in conference,
in our conference, yes, in lectures in English. So thank you very much.
And yes, thank you for organizing this. It's great, and comes back to the comment how to make closer clinician and researcher to clinic. Actually, the years was organized exactly for this, and small advertisement that it's happy to the Russian chapter is so active. And actually the best would be start just to be involved in this activity. And there you will find many clinicians and basic research scientists who are looking for today like progress in epilepsy studies. And it's great. Thank you so much also for that.
Yeah, thank you so much for organizing. It was great, and great job. And yeah, I think we should have more of these discussions and try to collaborate and interact more.
Yeah,
So
we will be very happy to such interaction and such discussion in future. Maybe not on the conference. We may have a discussion without Congress and so on, just like this.
Yes, and idea of brainstorming session, it's really nice idea, and we can have some topics in a month maybe, and try to do this because it's really nice idea.
Yeah.
Thank you. Okay, good luck with your best progress of the conference.
Thank you so much.
Thank
yeah.
you.
Thank you. Bye.
Bye-bye.
Goodbye.
See you.